And joining us now on the debate for the full hour tonight, in Calgary, Alberta, David Keith, Canada Research Chair in Energy and the Environment at the University of Calgary. In Boston, Massachusetts, Christina Yang, fourth-year graduate student at Harvard Medical School working in synthetic biology. And here in studio, Peter Singer, director at the McLaughlin-Rotman Centre for Global Health at the University of Toronto. His colleague Shane Green, bioethicist, also at the same McLaughlin-Rotman Centre. And Jerry Wright, professor of biochemistry and biomedical science at McMaster University in Hamilton. Good to have everybody on the program here in the studio and in Points Beyond, Boston and Calgary. I want to start by quoting from a magazine that is usually not breathless in its coverage of things around the world, but The Economist reports on the subject we are going to discuss tonight thus. Michael, if you would, bring this quote up. Craig Ventner, Hamilton Smith, and their colleagues have done for real what Mary Shelley merely imagined. On May 20th, in the pages of Science, they announced that they'd created a living creature. Like Shelley's protagonist, Dr. Ventner and Dr. Smith needed some spare parts from dead bodies to make their creature work. Unlike Victor Frankenstein, though, they needed no extra spark of Promethean lightning to give the creature its living essence. Instead, they made that essence a piece of DNA that carries about 1,000 genes from off-the-shelf laboratory chemicals. The result is the first creature, since the beginning of creatures, that has no ancestor. What it is and how it lives depends entirely on a design put together by scientists of the J. Craig Venter Institute and held on the Institute's computers in Rockville, Maryland and San Diego, California. When the first of these artificial creatures showed that it could reproduce on its own, the age of artificial life began. All right, let's go around the horn on this. David, to you first in Calgary. How big a deal is this?
I think it's a big deal. I think to claim that it's a bright line threshold that we've suddenly crossed is probably an overstatement. It's a fuzzy line. But there's no question that this is an important marker showing the enormous growing power of germline manipulation technologies that can be applied to microorganisms and eventually to people.
Christina, to you too. The first self-replicating synthetic bacterial cell. How big a deal is this as far as you're concerned?
I think it's a big deal. I mean, it's a big technical achievement, and I think it's captured a lot of people's imagination, which I think is really powerful. I think it's definitely a lot to say that it's actually synthetic life. I think it's a synthetic genome, so just the DNA code, not actually an entirely synthetic organism. And I think a lot of the sort of the tone of the Economist article and all of the news has been sort of maybe overstating that aspect a little bit. But it's certainly a big achievement in synthetic biology and very exciting for everybody.
Peter Singer, what's your view on that?
This guy, Craig Venter, is the Mick Jagger of biology. He just cut a platinum album, but he didn't invent music, and he didn't even invent rock and roll, but it's a big thing.
It's a big thing. You're making the distinction because he's got very good publicists. Is that what you're making? What you're suggesting here?
I mean, he is a bit of a rock star,
yeah.
And that's okay. As Christina said, he captures imaginations, and that's important. This isn't Adam and Eve. It isn't creation, but it's a major event in science.
Jerry Wright?
Yeah, I would agree. I think it's like the moonshot to me. It's a technological achievement that makes sense after everything that's gone on before it, and the world is not going to be the same after that.
The world is not going to be the same. You agree with that?
I would. I mean, I think it's tremendously exciting. I think it's a shame that some of the developments that have come before it didn't get quite the same splash in the press. Some of the work that's going on, for example, in George Church's lab to build synthetic ribosomes. So this is going towards building a synthetic cell, because as Christina said, this is what Venter has done, is build the synthetic genome, but not the rest of the cell into which he's put this genome. So
make the distinction there. What is it that he's actually done?
So he's built the DNA code. Okay. Mm-hmm. So each bacterium has its own genome, just like any living thing has a genome. And what he's done is to take the known sequence of that genome, the DNA sequence, which is in a computer bank, and he's added some bits and bobs to it so that they can distinguish the synthetic from the natural genome, and he's transplanted it into a pre-existing shell, if you will. So he's taken a bacterium, removed its natural genome, and put the synthetic one in. So I agree with Christina. This is not synthetic life in that he's created the entire life form from scratch. He has created the genome, though, and it's a major milestone.
And Christina, when they say it's self-replicating, What does that refer to?
That just means that it can grow like any other bacteria. If you just put it on a petri dish in the lab, it'll grow, it'll be able to eat the food in the petri dish and grow by itself. It doesn't need any extra help, any other organisms as a parasite.
And David, Craig Venter is, well, he's no stranger to genetics, he's no stranger to publicity, he's a pretty famous guy. Do you want to fill in some of the background on him for some of our viewers who may not know?
I may not be the best expert. I mean, he's been a real technical innovator, no question, in trying to figure out how to do much cheaper DNA sequencing, and was the one who kind of was one of the leaders of the race to sequence the whole human genome, and there was enormous hybrid of extraordinary development of computer techniques and biological techniques and biochemical techniques to pull that off so quickly. And he showed he had the organizational ability and scientific talent to put that together in a way that was really extraordinary. But again, as I said in the introduction, I agree with all the folks here, it's not a bright line that's been crossed, but this is a marker of something profound. And what's profound is the collective growing ability for us to directly manipulate genes of many organisms, and including eventually humans.
He is the guy, though, Peter, who cracked the code, is he not?
Yeah, so people will remember the Human Genome Project, the sequencing of human DNA, a huge milestone in science and biology. There was a public project and a private project, and he ran the private project, which sped up the public project. So he and Francis Collins, who's now the head of the NIH, National Institute of Health in the United States, were there in the Rose Garden at the Washington ceremony when the president announced that the human genome had been sequenced. So that's his pedigree, his background, if you will, a real rock star of science.
Did he get very rich thanks to that?
And maybe it's important to say, at the time, people were very, very skeptical he could pull it off. So the public project was a more kind of large-scale bureaucratic thing proceeding along a certain track, and there were pretty confident assumptions made that Venter couldn't possibly pull off what he claimed he would, and in fact, he proved them wrong.
David, do you have a connection to the discovery just having been transpired?
No, I have a connection to somebody who's a VP research there, and they needed a credible energy and environment person in Alberta when they set up a shell company. So I'm on the board of Synthetic Genomics Alberta, but that is really has no connection. I do energy and environment work. I'm not a biochemist.
Okay, you said no, but the answer is yes, right? You do have some.
Yes,
sounds like
yes, in a technical way, but that Synthetic Genomics Alberta has basically is a shell that might one day do something, and they wanted me because of my energy and environmental knowledge, because they were thinking about doing stuff in the Alberta world in that way, and I knew one of the people who runs that company, not Craig.
Okay, gotcha. Shane, tell me this: by doing this, coming up with this discovery, has Venter uncovered the essential genes for creating life?
Sorry, you said Shane. I'll jump in. No, he hasn't got the essential. He hasn't got the minimal genome yet. He is working towards that. It's something that he's been engaged in for some 15 years or more, trying to figure out what the minimal genetic complement is. This is not. He's not there yet. I would imagine that work will continue, but he's not there yet. He hasn't got the chassis, the genomic chassis
what's missing?
yet. What's missing is more understanding. We've got a lot of information. We've got a lot of DNA sequences from thousands of organisms now. But what we don't yet have is the Rosetta Stone. We've got the hieroglyphics. We don't have the stone. We don't yet know what all of it means. So it's going to be some time before we can figure that out, and from there derive that minimal genome, which is the minimal set of genes that an organism needs to survive on its own.
You say it's going to be some time. And Christina, I wonder, do you have a guess as to how far away we truly are from recreating a synthetic cell or a life form?
Oh, I don't know. I mean, I think it's probably going to happen maybe soon. I mean, and soon being 20 years, it could be still possible, but maybe even sooner. I think what's interesting about the idea of the minimal genome is that even a few years ago, they were working with a different species of bacteria that has a smaller genome than the one they ended up working with. But what ended up happening is that bacteria, because it has such a small genome, actually grew very, very, very slowly. So slowly that you wouldn't know if your experiment had worked for a month or so. And so they actually changed to a bacteria that had a little bit more DNA, a little bit more sequence to sort of help it along, be more robust, and that grows much faster. So I think the idea of the minimal genome is really interesting and can tell us a lot about what cells do and what they need, but I don't know if it's actually going to be really important for synthetic biology.
Okay. Peter, Craig Venter left, I guess what some people are calling a personal calling card on this. What is that?
Well, we don't really know what it is. That's the whole point. He put in what he's calling a watermark. But what you might think about, you know, when you go to the airport and you got your passport, and your passport has certain security features that you don't know what they are.
You can't see them, but somebody can see them.
Somebody knows. Mm-hmm. Well, that's sort of what's on the DNA here. So he's thrown in the names of some of the researchers, a few quotes, and a website that if anyone cracks the code, they can actually go to the website, and God knows what they're going to win. But that's all the watermark stuff. And the importance of that is that you can really quickly identify this organism as opposed to any other bug, and so it's really important to be able to do that for safety and security reasons.
Is that why he did this?
I think in part, yeah, in large part it is why people put these so-called watermarks in bugs, because you need to be able to differentiate these. It's just a security feature, like you got security features on passports, and it's all related to containment and security. That's a big piece of why people would do this. And what you actually choose to put in, your name, some quotes, I think that probably shows a little bit of his artistic flair. By the way, he's a sailor too. So he loves to go sailing, and here he's sailing in some uncharted waters. So tells you a little bit about his personality.
We're really torturing our metaphors on this program, I tell you. Okay, Christina, you wanted to follow up.
Oh, I was just going to say that the watermark is for certainly to sort of mark it as a synthetic thing in terms of security and sort of down the line thinking about future synthetic genomes and synthetic genes, but also experimentally, a much more simple reason for putting a synthetic watermark in is because if you don't have the sort of synthetic watermark that's different from the natural sequence, there's actually no way to tell at all whether or not your experiment worked or whether or not your plate was contaminated by the natural bacteria. So I think it's much more mundane when you talk about it that way, but I think that's definitely part of the watermark.
I like the kind of rock star metaphor better now that I hear it. Jerry, are other scientists now trying to jump on the bandwagon and do what he's done?
I don't know if there's a lot of people out there trying to build an entire microorganism from scratch. I think, you know, this kind of technology is right now incredibly expensive. So it's really only in the realm of a few, of a few labs that could do those things. You know, and why would you want to? I guess would be the big question. I think lots of people are really interested in building microorganisms that do specific things. Like you said in your intro, whether it's building better vaccines or making new medicines or what have you. But I mean, why would you want to go out and create a brand new organism, except that you've said that you've done it?
Because you can, yeah.
Because
exactly.
you can, and you can, but it takes a lot of money.
40 million so far, I guess.
Right. I mean, those costs are definitely gonna drop in the future, and that's really one of Venter's amazing impacts on science is that he almost single-handedly drove down the cost of sequencing genomes, right? To the point where it's almost trivial now to sequence at least microbial genomes. So those kinds of costs are going to come down, but there's so much diversity, there's so much microbial diversity, for example, on the Earth right now. Why you'd want to go out and build one from scratch, except to try and learn about those kinds of things. Which is really important. I think that's going to really do it. So then people will do that. But there's not a whole lineup of people wanting to build their own microbes because it's just not that exciting.
Got it. David Keith, you wanted to say.
Yeah, I want to echo that a different way. I mean, I think this is an example of sort of flexing of technological muscles and scientific might in a way that is enchanting in lots of ways. And this isn't about, as people say, that the need to actually or utility of doing this. It's about demonstrating how far we've come and what you can do. If we want to do something that's actually useful, making bacteria that make specific drugs or useful for energy production, or perhaps introducing, altering animals or human genomes, that's more likely what we actually are going to do. And in those cases, we wouldn't want to start from scratch. This is just a way to say, look at what we can do. Look at all the power we have. And that's really what's important, I think, is this is a marker of the kind of power we're developing.
Well, Christina, you're studying all of this at Harvard. Can you tell us what has been created via synthetic biology so far?
Oh, what other things have been created?
Yes.
So the big achievements so far have been it's sort of what people have been saying, making medicines and making fuels. There's been a lot of work producing a drug that's called artemisinin, which targets malaria, and the Kiesling lab at Berkeley in California has been working a lot on producing that drug and making it at very high quantities in yeast, while typically it's produced only in very rare plants. So it's sort of producing this medicine to help a lot of people. That's sort of the big triumph of synthetic biology before this. And then also producing fuels. So the people are trying to make biodiesels, different kinds of fuels in bacteria.
Shane?
Yeah, just on the artemisinin, and the other exciting thing there is that what they've done, part of the motivation is to drive down the cost. So as Christina mentioned, it's in a, you produce it from a rare plant. Even if you're producing it for $2–3 a dose, that's going to be too much for the need. So what the Kiesling lab is doing is trying to get that down to pennies, per dose instead. So that's part of the excitement around synthetic biology, is to do things faster, cheaper, more effectively.
While we're on the issue of excitement, are there a lot of students who are interested in this now, Peter?
Oh yeah.
Yeah, more than before?
Absolutely. I mean, and Christina is a great example. Synthetic biology is about bright, energized young people. And Christina can tell you about the competition they hold every year in synthetic biology. This is really energizing young people about science, which I think is a terrific thing. Synthetic biology may not be the fountain of youth, but it really is all about youth on the part of young scientists like Christina and others. Okay,
Christina, go ahead.
Yeah, so the competition that they're talking about is the International Genetically Engineered Machines Competition, or IGEM. And it started about 5 years ago with, I think, a handful of teams, maybe 5 teams in the US. And since then, it's grown exponentially every year to now there's almost 200 teams from all over the world. I think there's actually 10 teams from Canada and a lot of teams from everywhere. And the students work together over the summer designing a project that creates a new synthetic biology solution, a new design, and a new essentially a new organism. You'd start usually from a bacteria, usually E. coli, add new things, new genes into it, make it do something it didn't do before. And there's a lot of excitement at IGEM, and it's a lot of fun.
What have you done?
I'm too old to have been on a team because the first year he wasn't at my school when I was in college. But as a teaching fellow for the Harvard team, I've been working with the students to make systems where bacteria produce electricity, and this year we're working on actually engineering plants. So bacteria that can work together with plants to create new flavors in plants or different exciting things like that.
And Peter, do any of these student-led discoveries ever, lead to bigger things that have wider practical application for society?
Sure, absolutely.
For example?
Well, this is a young area and it's hard to give an example, but if you look at science generally, most of the folks that make real discoveries make them when they're young, in their teens, maybe in their 20s. And I bet you, and I haven't seen statistics on this, that with things like synthetic biology and these competitions, the age is actually going younger, because you got to be creative. You got to think about things totally upside down to make breakthrough discoveries, and that's where young people come in, and this synthetic biology is really enabling that. It's really cool.
Shane?
I just think it's important to mention here as well. I mean, we talk about Venter, and we see pictures of him and Hamilton Smith, and we forget that this work was actually driven by Daniel Gibson, a postdoctoral fellow who's done the doing in the work. Venter may be inspiring a lot of this, but I think it's important to acknowledge the young scientist, Dr. Gibson, who's actually taking this work forward, and many, many others like him that are often left out of these kinds of media reports.
David Keith, I appreciate that in our little universe here on this program tonight, there seems to be a great deal of positive backing and enthusiasm about the discovery that Venter has made. And while we understand there is no quote-unquote scientific community, it's too big and too diverse to just call it one thing, have you been able to gauge reaction either in your circles or in broader circles as to how this thing is going down?
Not this particular thing, but I've had a lot of conversations over the last few years about the implications of things that company and others are trying to do to engineer bacteria or plants to be large energy crops. So I've had, in fact, long debates over beers with Steve Chu, the current energy secretary, on that topic. And I think there's a very wide spectrum of views. I mean, in my view, technologies don't sort of come pre-packaged as good or bad, so I don't think that's a meaningful question. But I think there are lots of ways in which the impacts of these technologies are likely to be profound. I think actually most likely not in energy, most likely in genetic engineering of humans.
Do you think governments are paying enough attention to the point where they may want to get behind this with some more taxpayer support?
Oh, there's a huge amount of taxpayer support. I think the harder issues aren't taxpayer support. They're how to manage the social impacts and risks of new technologies. I mean, it's just a fact of life that technology tends to move faster than our ability socially to absorb it and our ability to regulate it. So, you know, parents all over the world sacrifice just anything to put their kids through college. They'll work night and day and live cheap. If there are technologies available that allow your kid to be healthier or smarter, and it's pretty clear those technologies are going to come, I'd say in the next half century, and that's probably a lot sooner. You're going to be able to do this for people, even in a limited way. It doesn't really matter if you ban it in a few rich countries, people will do it. And I think the implications of that for the world are pretty profound. I don't think there's a simple answer about whether it's good or bad, but I expect it's something that is going to be a central topic of debate and maybe even warfare over this century.
Well, let's get we don't want to do warfare here, but we are going to debate that here. You've segued us nicely into our second part of our discussion today, which I guess we're calling Cynthia and Pandora, because I guess we are going to open a bit of a Pandora's box here. Shane, why don't you get us started on this? The ethical considerations around Cynthia.
Sure, and I think David's started us nicely. I think the big question is, how do the potential benefits stack up against potential risks? So what are the potential risks of harm? And there's a recent report from some scholars out of the Hastings Center that sort of put the harms in two buckets. You got physical harms and then non-physical harms. Physical harms might be harms to humans or other organisms, to the environment, from the intentional or inadvertent release of a synthetic organism. So you've got those, and then there's other potential harms that are not physical in nature. Considerations of fairness, of equity of access to the tools of synthetic biology, the outputs of synthetic biology. And then even questions, David's talking about, modification of humans. So there's profound questions to be asked there as well, asked and discussed around, are there things that perhaps we should not be doing? And I'm not sure what the outcome of that dialogue is going to be, but I think it's worthwhile to have that dialogue at this nascent stage of the technology.
This is the should we be playing God question. Is that putting it
for lack
heavy?
of a better word? Yeah, sure.
Yeah.
Sure. I mean, that's often used, and I understand why it is. I think it's again, I would sort of come back to potential risks and benefits. I think trying to put labels on it, like playing God, tends to polarize the debate in ways that you're actually working against the dialogue. I'd like to see people come together and say, well, what are the potential harms here, and how could we act to mitigate those?
Well, let's do an example here. Christina, could you give us an example? Shane just mentioned an environmental problem. Give me an example how this could turn into an environmental problem if risks are not managed properly.
I think things that people worry about is things like if there's a antibiotic resistance gene that is in a synthetic organism and that gets out into the environment and that gets passed around, like perhaps you could have a lot of antibiotic resistance spreading through bacterial communities. There's also things that, a lot of things being discussed around GM crops, similar ideas, sort of biodiversity, problems of biodiversity, problems of
now, when we talk GM on this program, we're normally talking about General Motors, but you mean genetically modified, right?
Oh, genetically modified,
okay.
yes.
You got to watch the jargon here tonight.
Sorry.
Okay, understand. No problem, no problem. Keep going.
But yeah, I think though it's important to sort of know, I don't remember his name, but he was saying that technology moves moves faster than the questions that we can ask, and sort of moves faster than society can cope with them. And I think oftentimes our imagination gets ahead of where the technology is. And I can say for sure it is very, very difficult to do anything that changes the way a human cell works, even a cell sort of in culture, not within the context of a larger organism. Nobody is working on on modifying humans that I know of, and it's something that I think is really important to discuss, but I think has already been banned, and for very good reasons, and I think that it's important to talk about things that are the sort of realistic technological problems that could arise.
Okay, fair enough. In which case, Peter, let me follow up on that aspect of it then. She mentioned that you could get into a situation where, yeah, stand by, David. I'm going to put one to Peter here for a second. Who's paying attention, or whose job is it to make sure that we don't find ourselves with a whole bunch of antibiotics which are now useless because we've somehow played around and messed up?
It's a really good question, and in the first instance, it's the scientist's job, and actually Craig Venter and all those folks took a lot of steps to make sure that this particular Synthia organism doesn't escape. For instance, it can only grow on a really enriched type of substance, so as soon as it gets out of the lab, it dies by itself. The way I would cut this is you've got two types of problems. You've got so-called bio-error or bio-bungling or mistakes, and then you've got people with bad intentions. And just to give you a little story, on the weekend preparing for this program, I thought I would go and look up the report that the Venter Institute did or participated in on biosecurity. So on Sunday, I went to their website. Guess what? Their website was down. Today it was up. People make mistakes, and
okay, why was it down? What did you infer from that?
Well, what I inferred from that is that even in simple processes like websites, if you can make a mistake in your website, you can make a mistake in your Synthia. Now, you put a lot more attention into that, but I think it is important to say that bio-bungling, bio-error is a piece of the puzzle, is one of the risks, and we need to protect against that by having regulatory regimes, and really most importantly, a culture on the part of scientists that protects against error through vigilance, and that protects against the much, 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 much rarer episode of evil intention, which we could talk about. But I want to emphasize this is 99.9% good, and we need to think about those things and protect against them. And as one of the other guests said, actually be realistic about risks, but also be focused.
David Keith?
So two things. I mean, one is I think it's important to talk about enormous benefits. So despite the kind of strange reaction to genetically modified crops in Europe, so far, the genetically modified crops seem to have had enormous environmental benefits because they've greatly reduced pesticide applications, and most environmental scientists would agree, at least so far, the benefits have been pretty big and greatly outweigh the disbenefits. I want to really disagree with the claim that many people in the community make. I think it's so they can be safe, that nobody's thinking about manipulating humans. So first of all, in many ways it's already happening. There are a large number of babies living among us who were born by pre-implantation genetic diagnosis. So these are people who were IVF babies, in vitro fertilization babies, where a number of eggs are prepared, and then you do genetic analysis on those eggs and decide which one you want to implant. This is already, I mean, I think the first one was 20 years ago or something. This is now widely practiced. The idea that you could begin to use more sophisticated techniques to do that diagnosis, or the idea that you could actually do manipulation, is certainly not outside what's possible. Many people who are leaders in this field agree with that. They just don't like to talk about it in public.
Christina, you want a second chance at that?
That people aren't going to do that. We're saying in U.S. labs that certain things like that are off the table. That just means it's going to move to China.
Okay, let's get Christina.
I mean, it is true. I mean, IVF is certainly, there's a lot of ethical and very interesting and important ethical discussions to be had around reproductive technologies. Synthetic biology as a field, sort of that's been developing over the past decade, doesn't really deal with that. So I think maybe we're having two different discussions. So I think
I think we're using synthetic biology as a marker for the broader progress in genomic technologies overall. So I'm certainly not claiming anybody's going to make any synthetic humans, but I think in this conversation, maybe that's where we'll meet in the middle here. We've been talking broadly about using this event in synthetic biology as a marker of the enormous progress we're making in genetic science overall. And I think that science certainly does have applications to people. And I'm not making any ethical claims one way or the other. I'm just observing that people will do enormous amounts to make their offspring more likely to succeed, and that if there are technological ways for them to spin the dial, they're going to do that even if you pass laws against it. I think that's just a pretty realistic forecast based on technological improvements and human behavior.
Christina?
I think that's true, and I think that's something that we definitely need to talk about. And I don't remember if it was you who said it or somebody else about the sort of fairness and equality of access. I think that's going to be a really huge part of this discussion in terms of ethics. You know, yeah, and I think that's going to be a really important thing to talk about if we're talking about.
Sherry Wright.
Yeah, I think it's really important to get the nomenclature right here. That words are really powerful, and synthetic biology is no different than all other kinds of technologies that are being used in the life sciences, and I worry that we're going to end up demonizing a really important area of science that's emerging because of these concerns. These are concerns that are all part of life sciences that we have to be cognizant of, as was pointed out, and it becomes incumbent on us as scientists who know the nomenclature to make sure that we don't spread fears. The kinds of issues, so for example, we have a zebra mussel problem in Lake Ontario. That's an invasive species that is a natural species. No one designed that from scratch, and it's done all this terrible damage. So the issue of being worried about something synthetic escaping and causing some problem really is something we deal with all the time. We have dandelions all over our lawns here in Ontario because we're not allowed to spray them anymore. And these dandelions aren't from North America. They're from elsewhere. So I think we really got to be careful about this language.
Okay, let me try this with you, Shane, because clearly we're talking about a continuum here. You've got genetically modified plants over here, animals maybe over here, humans over here.
Right.
Are you concerned that we're starting to kind of play around in this part of the continuum a little too much?
No, not so much. I think you know I'm.
Because that'd be different. You would agree that genetically modifying humans
I would completely agree that would be a game changer, and I'm sympathetic to the concerns David raised about IVF and saying we are at a point where we're not manipulating, we're selecting. So that's an important distinction to be clear about the language. But I think the important thing is that we're at this is the perfect time to be having these conversations. All too often in the past, these kinds of dialogues have happened after something has come out and we've reacted. Look at Dolly the sheep.
Yes.
They cloned Dolly the sheep. Suddenly everyone's up in arms, wondering what are we going to do? How are we going to respond from a policy perspective? Scientists were saying, "Oh, kudos, nice work," but we expected it. It was the next iterative step.
So try to stay a step ahead of the process.
Well, try and stay a step ahead by having that dialogue. And synthetic biology comes along at a time where I think that dialogue between scientists and social scientists, policymakers, and the public is increasingly, it's present, right? We're encouraging, some scientists are taking it on themselves. You see in the Venter paper that they acknowledge the ethical issues and they're engaging in the debates. Some funders insist that scientists, to get funding for these kinds and other kinds of research, work with ethicists, with lawyers, with environmentalists to proactively consider and address these kinds of issues. So I think it's a good time to start having those conversations while it is still a bit sci-fi.
Gotcha. Peter Singer?
I was just going to say, I think there's something concrete, Steve, that we can do, because these discussions actually happen a lot more in the United States than in Canada. But 18 months ago, I wrote an op-ed piece in the Globe, and the title was, "Is Canada ready for bioterrorism? Is Canada prepared for bioterrorism?" And I was talking about the advances in the life sciences and how well we're dealing with them. And my answer at the time is probably, but we don't know. In the United States, they've had about 5 or 10 studies, the National Academies, Congress, the Venter Institute, looking specifically at advances in the life sciences, what scientists and others should do to mitigate those risks. That's a dialogue we haven't had publicly enough in Canada through institutions like our national academies. And that's something I think actually that we should do in Canada. I think we probably are pretty well prepared in terms of the intelligence and law enforcement communities.
Well, my guess is when you wrote that, I remember reading that piece and I remember thinking, boy, there's going to be a lot of people who are probably going to call him today saying, you shouldn't be talking about this, right? Did you get that kind of feedback?
A little bit, but we have to be talking about it because I totally agree with the points people have made. This is 99.9% good. This is about helping the billion people who go to bed hungry every night. It's about saving the lives of the 800,000 kids who die of malaria every year. That's what this is primarily about. But the flip side is there are some risks, either unintentional or much more rare intentional. And we ought to be thinking about the realistic risks, anticipating them, and actually engaging scientific communities in being alert for them through institutions like the Council of Canadian Academies, our national academies. And all I'm saying is, we've been talking about flu for a long time in Canada. The US equivalents have been talking about bioterrorism. There's a bioterrorism academic center on every corner, not exactly, but I can't think of one in Canada, and I don't think we should go to that extreme, but we should start to look at some of this stuff. Why? Because that leads to action in terms of mitigating some of the risks in something that's overall positive.
But I guess, David Keith, it's fair to say that, you know, throughout the history of science, our morals have always had to play catch-up to our technical capabilities. Isn't that fair to say?
Oh, completely. And I'm not advocating that we should stop the science. I think I agree precisely what the last speaker was saying. I think it's important to get the conversation going early so we have a better chance to manage it. The last time I was on this program, I was talking about this work that I've been involved in engineering the whole planet. And a lot of people think I'm reckless for talking about it. And now it's become something that's quite widely talked about. And my view has not been that there's a necessary answer to what we should do, but that it's important to talk clearly about what our capabilities are so we can make better decisions. I do think that you may be hearing some sort of downplaying of the potential risks of malicious use. You know, people were talking about it's just like invasive species or dandelions. I often hear people in the genetic community say, you know, we've always done crossbreeding. This is no different from regular genetic crossbreeding of plants. Yet at the same time, they say they're going to engineer rubisco, the key enzymes of photosynthesis, to make it more efficient. I don't believe both those statements can be true. If we're really able to do the latter, to actually re-engineer photosynthesis to make it a lot more efficient, which I'm not sure if that's true or not, but if it is true, then the implications of free-living organisms are obviously pretty extreme. And the possibilities of warfare are pretty real too. It's important to look carefully what the Soviet Union did in its dying days in the biowarfare department. It was the only time an advanced state really worked really hard on these topics, and the results were pretty horrifying, and that was 15 years ago.
Christina, let me follow up with you on that. Aside from Ventnor's lab, synthetic biology is, I guess it's a bit, well, I don't know. Some of the stuff I've read suggests a bit Wild West out there. Scientists, students, a lot of experimentation going on. I know Peter says that there's a 0.1% probability that there's something evil or malevolent happening out there, but you know, mistakes can be made, right? And I wonder, in your daily activities, what kinds of protocols do you go through to make sure that mistakes, I'm going to assume you're not part of that 0.1% that's malevolent and has evil intent, but how do you make sure that mistakes aren't made?
Well, all academic labs and industrial labs have a lot of regulations and a lot of oversight and as to how they take care of bacteria and any other or any organisms at all, and how they deal with genetic recombinant DNA technology that's been going on for about since the 70s, and how sort of all these rules in sort of how people use where they can use it, what kinds of labs can have different things. There's a lot of regulations in place. I'm not saying that that doesn't mean that there should be more regulations or different kinds of regulations for new technologies, but there's certainly a lot of ways that things are safe when we use them in our lab, for sure. And that doesn't mean, again, that there won't be a mistake, but we are very careful about where things go, you know, how we can dispose of things, how we take care of everything.
And again, tell me how it works in your world, because if I understand the process properly, you guys you're very open about things, right? You make a discovery, you post it on a kind of like an open source wiki online so that everybody else has access to that information. Is that how it works?
That's sort of the idea. It certainly is for iGEM, for the competition. If you present a thing that you made, a genetic sequence that you made, that you designed, which is usually a combination of different natural sequences, if you make that sequence, then you have to share it with the whole iGEM community. So things are very open and there's a lot of talking about access to the DNA and sort of sharing of all these things. In the broader community, things are a little bit tighter, but still very open. You know, we wait, I guess, till we publish a paper before we let anyone else use the DNA that we've made, but that's the same idea. We do like to share between the community, between the different labs, to sort of develop new and open and accessible technologies.
Yeah, Peter, I was surprised to read that because, you know, most of what we hear about scientific breakthroughs, the information is very tightly held. It's all extremely proprietary. We know right now on gene patenting, for example, we had a program about this about a month or so ago. You know, it's extremely tightly held, and One company still going all the way to the Supreme Court to try to fight to hang on to their exclusivity on this, much to the consternation of some women who would like a second opinion when it comes to figuring out if they've got BRCA1. So how much of this open source stuff is really going on out there?
A lot, and it's a really good time actually to compare nuclear with biological threats. Because when people think about terrorism, about warfare, they're using the nuclear metaphor. Nuclear was born classified, it has huge capital costs, and it's very centralized, so it's going to be likely state sponsors of nuclear terrorism or nuclear war. Biological was born unclassified. It's totally decentralized. You could basically do it in your backyard in some cases. And so it's very open, and that's a good thing, and it's going to stay that way. There's not a single choke point. And so this openness is actually really important also to protecting against threats. For example, we've been talking about bioterrorism. Imagine a bioterrorist attack that you need to protect against with a vaccine. And that's called a countermeasure. This sort of biological breakthrough, synthetic biology and others, we talked about its role in vaccines, can also help in countermeasures. So on balance, since there's way more good guys out there than bad guys, if you will, on balance, it's a good thing that biology is so open, but it requires different ways of mitigating risks.
Only takes one bad guy to kill 100,000 people, though, right?
It takes one bad guy who succeeds.
Right.
And that's why, you know, the defense team has to be right every time. The offense team has to be right only once, which is why it's really important to think about the risks, to outsmart the adversary, if you will, and have those measures in place. But that should not lead to a closed system, because that'll put us worse off.
Well, having said all that, Shane, I gather the Obama administration is going to spend the next 6 months investigating all of this? What do you think they're looking for?
I think they're trying to get their heads around the implications of this. I think security will be a big issue, a big part of their debate, their discussion. You know, in the wake of the anthrax attacks in 2001, I think it was, you know, you saw a real upsurge of discussion around mitigating risks of bioterrorism. You know, and there was a meeting of the national academies talking about making pathogenic genome sequences openly available, to come back to what we were talking about. And they decided, as Peter was alluding to, it's better and better, it enables better defense to make it openly available, so that those who would seek to develop those countermeasures and treatments and such can do so more rapidly and more openly. So I think they'll probably be sort of thinking, looking back across what's, you know, those discussions over the last 5 years or so, and then turning to synthetic biology and asking questions like, what is new about this? Is this different, or is this just the next iterative step in genomics and nanotechnology and engineering? And, you know, what questions, if any, are new here?
Well, we've got about 7 minutes to go here, and let's see if we can put a few new questions on the table in our home stretch. Here's an excerpt from New Scientist magazine, an editorial by the aforementioned J. Craig Venter, Clyde Hutchison III, and Hamilton Smith, all of whom are known to those of you around this table. We now have the means to design and build a cell that will define the minimal set of instructions necessary for life, and to begin the design of cells with commercial potential, such as fuel production from carbon dioxide. We can assemble genome-sized stretches of DNA that can also be used to mix and match natural and synthetic pieces to make genomes with new capabilities. This speaks to what I guess all of you have been saying, David, I guess you in particular, that this is 90% plus overwhelmingly a positive story. So what would you like to see, David, to you first, what would you like to see this new technology used to create?
Well, first of all, I want to really wholeheartedly agree with the earlier statements about the importance of openness and the way this is very different from the nuclear security world. And I think that's terrific. I don't think it means there's no risks, and I'm not saying anybody else was saying that, but I think all else equal, an open world is a heck of a lot better than a closed world. I mean, I hope this is used to better human welfare. It sounds very airy-fairy, but I don't know what else to say. I mean, I think these technologies, I think, are particularly potentially important for the welfare of the poorest billion, to deal with the health concerns of the poorest billion and food and energy for the poorest billion. That's where I see the most excitement. And part of the reason is that these technologies require enormous, well, not enormous, they require what seems like a lot of money to do the actual science, but then because they're self-replicating, they're things that you really could spread around the world pretty cheaply. And so as a way to really help the poorest billion folks who don't have access to electricity or sanitation and so on, I think this is really terrifically exciting, and that's what I hope for.
Peter Singer, the bottom billion, what would you like to see?
Totally agree with the bottom billion, because innovation saves lives. But let me take a concrete example that helps both the bottom billion and Canadians. Remember flu, H1N1?
I think so.
Remember the problem with flu, which was they isolated the virus in the spring, and it was about 6 months later in the fall that the vaccine became available, and the shortages and the lineups and the criticism even this week. You know why that was? That's because we're using a 75-year-old chicken and egg technology to make that flu vaccine. And one of the things that synthetic biology could enable is much quicker vaccine production. Imagine having a flu vaccine not 6 months after you get the virus with all those lineups, but a week later with no lineups.
But here's the follow-up question.
And that is what this can accomplish.
Even if you had it available a week later, would 75% of the people still refuse to get it, as was the case this time?
Well, you know, I think that more people would get it, because the reason people didn't get it, and that's what these reports are showing this week, is all the chaos and the lineups. I mean, if you see this huge lineup going around the block on TV at your local vaccination station, you might not go down there either. And that's the scenario that really bugged people, and that's the scenario you could actually prevent with science by using a new technology to develop vaccines much more quickly, rather than having to wait 6 months for a 75-year-old chicken and egg technology to kick in.
Okay. Jerry, what would be on your, I mean, we've got an example here of how we might help the bottom billion. There's environmental concerns. What would be on your list?
Well, I think maybe after this conversation, the first thing I would do is try and give everyone a lesson in biology and genetics. I think that there's a tremendous amount of it. These technologies are going to be with us for a long time, for the future, and I think it's really important, as was said, we have to get the dialogue going with the people who are going to fund all this and the people who are going to purchase these technologies. And you know, the way it can blow up on people, like has been seen with the GM crop issue. So there's a tremendous. I would love, first of all, I would love us to be able to have a really well educated and well thought out response to all of these things, as opposed to sort of knee-jerk concerns.
Okay, I want to save a minute. I've got 2 minutes left, and I want to save a minute for Christina on her wish list, and then finally a minute for Shane on his wish list. So Christina, go ahead. Synthetic biology leads us to what fantastic new development that would excite you?
I agree with everyone who's been saying all the things people have said before. I think those are all going to be happening soon, hopefully, and those are things that I would love to see happen. I think what's really powerful about biology is that you can do things that grow themselves, that can adapt to changes in the environment, can do things locally with less energy, using less materials, and so fewer materials. So I think that's going to be really powerful and really important to getting us energy independence, all those things, and that will help everyone, I think, in the whole world. So that's what I like about synthetic biology.
It's basically as broad as your imagination can take you. It sounds like what you're saying.
Exactly.
Okay. Shane Green, you get the last word today.
Oh, fantastic. I mean, one place my imagination takes me is bioremediation. You know, we've made a right mess of the environment, and this is a global issue. This is not limited to developing or developed countries. We've made a real problem out there, and through synthetic biology, I think there's real potential to develop new bacterial or enzymatic approaches to cleaning up some of that mess. I mean, look at the oil spill now. Wouldn't it be great to have an enzyme that could catalyze the natural breakdown of those products before they get to the shores, before they get to the seabirds and the corals and the like?
Do you have any idea how soon that might be a reality?
No, I don't, but hopefully my colleagues that are in the lab still might.
Peter, got a hint about, you know, when we might be able to look forward to that? So every time there's an oil spill, which sadly is too frequently, we don't completely give up hope on ruining the environment for the next 100 years?
Tomorrow would be nice.
Tomorrow would be nice.
It's not going to be tomorrow, but you know, a realistic goal would be the next time. Or a bug that does create oil, so you don't have to drill it out of the ground in the first place.
Christina, you want the last 20 seconds?
I was just going to say that that actually already exists. I think what the first organism ever patented, the first genetically engineered organism could eat oil. And I think there was a lot of problems in getting it actually into the environment and releasing it in a way that, that people agreed with. So I think that things are going to change a lot, and maybe we'll be able to develop safer and more better ways to eat up the oil in the future, hopefully, and make oil ourselves.
A good, hopeful note to leave things on. Can I thank our guests out of town for being with us today? David Keith at the University of Calgary joined us in our Calgary studios. Christina Agapakis, the Harvard Medical School, joining us from Boston. Thanks so much to you two. Jerry Wright, McMaster University in Hamilton, good to have you on the program today. Shane Green and Peter Singer at the McLaughlin-Rotman Centre for Global Health at the University of Toronto. That's a lot of firepower around the table today. Thanks so much, everybody.
Thank you.
Thank you.